so yeah, now I'll talk a little bit about the types of properties that you can compute with structural information and how you measure those. And here's just a table of a bunch of different protein properties that may be important or that are important in terms of a living cell. So on the left here are just some basic properties that we'll talk about. But you can get into detailed enzyme kinetics and activity at certain temperatures, different pH, stability at, again, different temperatures, different pH, specific enzyme rates, reaction rate for a substrate that you're interested in, and how efficient your protein, your enzyme is. And a lot of these properties are important in terms of protein engineering and strain design. And like if you want to have a high yield of your chemical that you're making in your metabolic engineering process, you're going to want a very efficient enzyme that's stable and very specific and produces a high yield of your chemical. And so these are very important things for protein design people and engineers to consider in their research. So again, there are experimental methods to get a lot of this information. You have enzyme assays to tell you the rates of reactions. You can study the effect of a mutation on a protein and its structure as well as its function. And as I mentioned, you can use this information for protein design. There's this whole field of proteomics, which tells you how much of a protein is in your cell at a certain point in time or at a certain condition. There are a lot of studies on protein-protein interactions. So proteins not only interact with small molecules, but they interact with each other, and that can cause a lot of things to happen. And so their experiments determine what those other interactions are. And finally, there are a lot of studies on protein-DNA interactions. So proteins, like transcription factors, will regulate functions in your cell to either reduce or increase the expression of certain DNA transcripts. And so the experiments here that you'll see mentioned a lot are ChIP-ChIP, ChIP-seq, or ChIP-exo experiments. So you can do a lot of this stuff experimentally, and then you can do some of it computationally as well. So as I mentioned before, you can predict a structure with homology modeling. You can compare two structures just as you can compare two sequences with sequence alignments. You can compare their 3D structures in 3D space. You can calculate some of these interactions of small molecule binding or protein-protein interactions with a method called docking. And you can simulate movement and activity with some really advanced methods, which I'll also mention in this lecture. And so here's just kind of like a scale of how accurate and how expensive computationally these methods are. And so it's kind of laid out as well here, but these methods really, I don't think, can be applied at a genome, especially like QMMM cannot be applied at a genome scale. That tells you things like that actually simulates a reaction happening with the hydrogen, with the bonds changing and stuff like that. So that's a very computationally expensive method, but very accurate. But in terms of using it for a whole cell to simulate all the reactions at once using the structure, that is still probably years off from that. And here's just a diagram of all these properties and what they look like on a structure. So an easy way to think about these properties is how they're calculated. And a lot of them can be easy to understand because they're really just geometric calculations. So these are things like calculating secondary structure. All they do is they measure potential, they measure distances of potential hydrogen bonds. And if they fit the constraints that they put on, say, an alpha helix, which should have a certain length for every hydrogen bond, then it's assigned a secondary structure of an alpha helix. You can do cooler things like calculate where a tunnel is in a protein to understand how a small molecule may enter a binding site, and if a drug may be too large for it or something like that. And you can calculate the surface and volume of proteins. using different methods, and that will tell you how buried or exposed a certain residue is. That will tell you how large a protein is to fit inside a membrane, to understand how much space it's taking up. So these are simply geometric calculations, and if you're interested in visualization, that kind of launches you into this whole other field of computer visualization, because all of this needs to be visualized. And that's a whole other topic, I think. But it's really interesting. So I'm starting to work a little bit on that kind of stuff in 3D space and even like VR stuff, and it's really cool. So next, you can calculate a lot of what we were talking about in terms of amino acids and their properties, and understand how they're distributed throughout the protein structure itself. So that will tell you like what kind of environment it likes to be in. So the easiest example to understand here is that membrane proteins will have hydrophobic residues on their outside, while cytosolic proteins will have hydrophilic residues on their outside. So these are basic physiochemical properties that you can use to understand what a protein looks like and where it fits into your cell. You can compute things like thermostability and certain like ionization state under different pH conditions, and these can tell you a lot of different things. So an application we'll talk about in the next lecture is how we've used protein-specific properties like thermostability to compute a metabolic network under different temperatures and what that can do to the network itself. So these last 3 slides, I'll just give a brief introduction on some of these more complicated molecular computations that have been developed. So one that has been worked on a lot is docking. So docking is the prediction of a small molecule binding to your protein. And this is widely used in the pharmaceutical industry because what they'll have is they'll have probably thousands or tens of thousands of potential pharmaceuticals, and they'll want to understand, do they even bind to our protein of interest? And they'll use docking simulations to understand that and rank them based on that to either get their billion-dollar drug in the end, or hopefully avoid the billion-dollar clinical failure, which can also occur. And this is pretty useful because it's a decently fast algorithm compared to the next 2 things that we're going to talk about. So next up is understanding the dynamics of a protein, and molecular dynamics is the main method used here. And this allows you to compute movement. And what they do is they represent each atom, or in some cases each residue. It depends on how detailed you want this representation to be. And they represent that atom and simulate its forces in terms of its neighbors around them. And so this is really just calculated molecular simulations based on first principles using like Newton's laws of motion. And you don't have to, this is not just applied for proteins, but also applied for material science as well. And here's just like, that was just a simulation of water melting, and that was a simulation of a carbohydrate binding to a protein. And with these simulations, you can get a lot of information. You can get, as we saw earlier, you can see how a protein folds and predict that. You can see the motion of different molecular motors and see how those function. You can understand the impact that a mutation may have on a structure, and so we'll also give an example of how we used MD on a couple proteins in the red blood cell to see how the mutations would impact the proteins themselves and then impact a metabolic network. You can see how active site residues are oriented in space and see if they are oriented properly to bind something of interest. And then you can calculate free energies, and so you can calculate things like what's the stability, what's the free energy of binding of my drug to this protein. However, one thing this does not do for you is simulate chemical reactions, as I mentioned before. And with that, what you want to do is use more detailed methods, which are called quantum mechanics. And this is not my field of study, and I really don't know much about it. But what they do is they represent, instead of an atom as a sphere in space that it's defined by forces and charges, they actually represent the electrons in their shell and how they interact with other atoms with electrons in their shells. And so this is a very complex computational technique, but it's very important. And it recently, in 2013, won the Nobel Prize for developments here. And what they can do is they can model proteins using molecular dynamics like this, but instead model the part of the, say, the catalytic site using quantum mechanics methods and actually simulate the reaction happening at the core there. So yeah, so this is very valuable information, and hopefully someday can be applied to a much larger scale. But it's really cool stuff as well. And so what I'll talk about next time is, instead of looking at these proteins one by one and calculating their properties and understanding them and per residue or something like that, what we can now do is we can extend this to the genome scale and think about proteins in a whole cell. And so here's one of these illustrations by David Goodsell. And the work that we've been doing is to extend the knowledge that we can and the information we can get from structures themselves into a whole genome scale metabolic model. And that's what we call a GEM Pro. And yeah, so that will be saved for next Tuesday. So thanks for your time. If you have any questions, just ask them, and I'll be happy to take them. So thanks.